Let me start by introducing Professor Xia. Professor Xia is a professor at the Department of Computer Science in Swansea University. He started his lecturing career as an RCUK academic fellow and has been an investigator on several projects funded by the EVSRC, Lever Hamleys, and so on. And he has been working on different areas in pattern recognition and machine learning, and also a lot in different areas of computer vision as well. So his recent works are about detecting abnormal patterns in complex visual and medical data, assisting diagnosis using automate image analysis, fully automate volumetric image segmentation, registration, and motion analysis, which is an area that I'm also working on, and machine understanding and human actions, efficient deep learning and deep learning on irregular domains. So Professor has published over 170 research papers and co-edited several conference proceedings already. So it has been a great achievement. So without further ado, I will pass the table to Professor Xie. So feel free to start. Thank you very much.
Thank you, Will. Thank you very much. Let me just open the slides first. As I said to you earlier, I'm really hoping that I can visit Durham in person this time. I planned this all along. And we discussed last term to arrange a bit later in the term, this term, to allow the in-person to happen. But it seemed turned very hectic for me. So in the end, we have to go for online version. So yes, I would take maybe 50 minutes or so to talk about some of the work we do in terms of healthcare applications. Now, the title has a keyword deep learning. I hope we don't disappoint it after the talk. Not much of those are perhaps not considered that deep models nowadays. So really, I'll just give some of the shared experience, some of the work we've done, particularly in the health informatics sector. And I'm certainly latecomers to that sector, and I'm more image processing person, perhaps similar to Toby in terms of background. So health informatics and particularly healthcare data analysis is much later to my own research area. But it's more, as I said, shared experience, and I'm looking forward to any comments and suggestions. That'd be very useful. So yes, so we are a group of many students in my lab looking at various different applications, but fundamentally we're interested in image analysis, segmentation in particular, but also registration, text analysis, and so on and so forth. We do a little bit of computer vision as well, some elements of 3D vision, but largely to do with, for example, real-time applications, but we have been working on it for a number of years now. Medical is being the focus for quite some time, and inevitably, as everybody else, deep learning is a factor in many of those applications and techniques behind all those. As I said, it's a reasonable size group, mostly PhD students, and working on various different funded projects, and some of them EPSRC look at a data privacy side of issue, how to analyze the data and open data settings and without compromise privacy. But much of those, again, project reflecting our key interest is really in the medical domain. And you can see they are mainly focused on medical image segmentation. So two things I'm going to mention today. So broadly in medical application or health. So I will start with an example of medical image analysis, which have some element of deep learning, but also to do with how we actually represent the models we are trying to extract. That has consequence in terms of model development as well. And then I'll talk briefly about two sort of medical data analysis. So entirely nothing to do with images per se, entirely to do with electronic health records and whether we can derive predictive models from those, and perhaps equally important, if not more, whether we can derive features and biomarkers from those. Can we interrogate the model so that produce certain features that clinicians can understand the model itself, and also predicted biomarkers as well. So hope that's okay. So any points, if you have any questions and anything in the comments, just interrupt me. So in terms of segmentation, I think just in case any people not working in areas, we're really looking at a relatively large volume of data. So each volumetric data you're looking at, so the smallest size you can look at is a 512 by 512 by 512. And then an anatomical structure that you're trying to extract often is quite small. So for example, I'm just going to move my screen a little bit. It's getting everywhere. So one example here, let's say we're interested in this area, which is the root of the aorta vessel, and you connect into, you can hardly see them, but there's a, I don't know if you can see my mouse cursor here. You can have the left ventricle, this blood pool. And let's say if you're interested in measuring the geometry of this connection between the ascending aorta and the left ventricle area. So you have 3 leaflets were placed here. If they have issues, for example, causing regurgitation because of the calcification build up at the leaflets regions, then you have to replace them. You have 2 options, broadly speaking. You can either open heart surgery, so that carries significant risk. Another way to do it is using catheter. You deliver artificial valves through the blood vessels and then place that into place, which carries a far less risk, but not as necessarily applicable on all patients. So you'd have to assess beforehand what to do. And part of the assessment in choosing the pathway is to understand what sort of geometry you're dealing with. And also, once you have the segmentation, you can also analyze what level of calcification occurs in certain regions and so on and so forth. So segmentation is very much the first stage, if you like, to understand the process and optimum pathway for the patients. And particularly for cases who are borderline with in terms of risk and benefits for the 2 different approaches, whether open hearts or using catheter-based delivery. So that's very much relevant. So segmentation plays a role in that decision making. So I'm going to briefly talk through the segmentation work, which may be interesting to some of you. So these give you in a snapshot what sort of process we're looking at. As I said, we are looking at a very small region outside multiple, hundreds of millions of potential hypotheses we have to evaluate. We narrow down very quickly and then find the region of interest and trying to segment them to the extent that we can consider acceptable in terms of accuracy with geometry and so on and so forth. And the soft tissues do look very similar. Whatever modality you use, this one is CT, you could use MRI and so on. So soft tissues do look very similar. So what do you do in order to remove the ambiguity as quickly as possible? Now, we are using the approach so-called interactive segmentation. We're saying that user plays a significant role in eliminating those ambiguities and localize the region of interest as quickly as possible. Of course, you can argue a different way to approach this, but we are saying that interactive segmentation would be quite an efficient way to evaluate segmentation as you produce results. Then comes the issue, if I'm interested in the geometry, so how to represent geometry and in what stage the geometry representation should take place? Should it be at the beginning of the segmentation or should it be at the end of segmentation? So broadly speaking, you can either perform segmentation and then let's parameterize the shape we have. That's one way to do it. Or you can start with parameterized model or representation and you say, let's iteratively find then until reaching the stage where you think is good segmentation, then you have representation that way. Both approach has benefits and its advantages and disadvantages as well. For example, the first one you have here, which is very commonly used, you do have difficulties to parameterize. It's not that straightforward as you might imagine. How to isolate individual regions and realize a single region of interest, and maybe indeed there's multiple regions of interest. So not that straightforward. So we are looking at in this particular work, looking at representation as well as how we carry out interactive learning online. So on the representation side, here's a very basic animation, just show you what I mean by implicit representation. So suppose in 2-dimensional space, this square you have here is your basic model, if you like, and that's your shape you're trying to capture and use that to localize the region of interest. So that's in 2 dimensions. So implicit representation basically means that instead of you parameterize this two-dimensional, the square we have here, you embed it into a high dimensional space. So it's one dimension, you embed into 2D and visualize 3D. And we can sort of change the embedded function. In the process of doing that, you have a deformation of your two-dimension original representation of the shape. So you are going to slightly higher dimensional space and evolve according to whatever classification, for example, in segmentation case, give you, and you drive that deformation and give you the segmentation you need. The trouble, however, is that you don't have explicit control over the, in this case, in the two-dimensional space, the contours. So if I want to manipulate them, say merge them, or just want to change the shape slightly, then nothing particularly can control them because you have to manipulate through this implicit function. Okay, so what we're saying that instead of using purely implicit representation, how about merge the two using semi-implicit or parametric implicit representation? So we have control points around two-dimensional surface we have here, and then we be able to control the representation as we evolve it implicitly. So many different ways you can do that. In this particular work, we're saying that let's using compactly supported radial basis function to do that, and you can localize them non-uniformly in the region, and you can parameterize them in that way. So I'm not going to go to the mathematics in much detail. I'm just going to show you some of the key concept ideas. So you could have, for example, uniformly distributed BSP lines put in a place, and then you can use that to approximate the shape of the embedded function. So in the simplest case I'll show you there, it's a cone-like shape. Now you can use this to uniformly displace them. Now the problem, one problem is that the geometry you're trying to capture may have locally have different complexity level. So the dummy example we have here is the bunny shape we have here. You can see regions have a high curvature, and then you have regions with low curvature, smooth changing surface compared to sudden changes, which you do wish to capture. So that entails either you use denser representation to start with, or use sparse one with a risk of some places you're not going to be able to capture details. So in this work, we propose, instead of doing uniformly, choosing either dense or sparse, you can do that by analyzing the complexity of the shape itself. You can approximate what's deformation happening at the moment, and then you can adapt to changing that. So there's few tricks how to integrate in that, allow that to happen. So we're using wavelet to analyze the complexity as we evolve in the shape, and then we change the representation in that particular form. Now, once this is sorted out, then we are ready to say, okay, we can deform them now. Now, in a conventional sense, when you try to deform those implicit representation, you're looking at solving the PD problem. But because we have control points for those parametric representation, so we can in fact transfer that into an ordinary differential equation, and that makes things a lot easier. And then you can choose whichever way you want to segment them. You could say, let's using a piecewise constant model. We're saying that certain statistics inside the region is significantly different to that outside the region. Then that statistic could be derived from your classifier. So you could have deep model, whatever that is, to produce certain numbers, the probability, for example, indicating the region of interest. So then you can use in a very simple, basic piecewise constant model to indicate the partition between the two. So this equation basically illustrates the concept of that. So you could have C1 indicating what you're expecting inside, and this one indicating what's outside, and you want a separation between the two, which is based on whatever deep model produced as indication being the region of interest. You can then impose certain regularization on the model, for example, a smoothness as one. Then all that taking place, you're ready to perform the segmentation in a deformal sense. I didn't get that. Could you try again? Apologies, I don't know why serial being activated. Nice. Okay. So what I said, you can use any kind of deep models or any classifiers to indicate the foreground and background. And the one thing which I briefly touched upon is that the number of hypotheses you have to evaluate in order to detect a very small structure in the scans you're dealing with is enormous. So if you said if you have 512 by 512, 256, one with the axial dimension, you're looking at over 67 million possible regions to evaluate. That's clearly not feasible doing the full 3D evaluation. So what we did is that let's divide them into 2 stages, have a very basic naive Bayesian-based approach to say, let's classify into 3 different regions and quickly eliminate those ones which we don't think will be containing the objects of interest. So this diagram here just showing that on different regions were showing different probability. You do see good separation of them, and then you see overlap as well. But the objective of this one is a quick elimination. You get rid of majority of the 67 million voxels to say that's not going to be anywhere near regional interest, and then the rest of them you can pay a little bit more attention to. So here again, this is where so-called deep model comes in, and by nowadays standard is nowhere near deep in a sense of the layers we have in the structure architecture of the neural network. So we look at very basic ensemblement of small convolution and batch normalization followed by pooling. But that itself, this basic model like this one we have here, is sufficient to tell the difference between, in this case, the aorta region and the surrounding tissues. And with the help also, the shape representation also give you constraints in terms of what regions you're looking at. And a few things we also do, as I said, batch normalization and feature aggregation, and feed them back into the classifier, which in the neural network later stage also helps to derive useful features. So these are some of the tricks I think lots of work, neural network using nowadays as well. Right, so let's have a look at putting all those together, what results we have. So that's the top row we have here. It's if you use everything else the same, but just using standard uniform representation with our proposed model and classifier, you get a reasonable localization accuracy. You know, they are at the right region, so that process is fine. The problem is when you're trying to analyze how closely they are to localize the region of interest, and then they're starting to show the difference. So ours is more accurate in terms, these are the 3 leaflets you can imagine on top of those, which ours is much more accurate in terms of the segmentation. And here's another example. If you're indeed interested in ascending aorta, then that's what sort of results we're expecting there. And you can see the quick convergence from naive Bayesian classification. You eliminate most of the regions, and then quickly go into, in a few iterations, you can find of the deformable objects you're looking for. Right, so I think that gives you hopefully a little bit of flavor of what we do in terms of the segmentation. It really merges some of the things we've done quite some time and deep learning. But the model, I said, reiterates that they are very shallow compared to what deep models we're referring to. I don't know if any questions this part, any comments. Feel free to shout out if anything at all. Right, okay. So I'm going to move on to what I said about analyzing health data and trying to figure out what biomarkers may be useful in assessing certain medical conditions. Well, I start with this slide, which basically tells everything. Now, the machine learning side or neural network side is very, very basic. We can go through that very quickly. But I think there's some findings may be interesting to you as well, if you're interested in the space here. So we are using a large data set called Cell Data Bank that captures Welsh population, about 3 million also, and dates back about a couple hundred years. The hospital admission data, most of them are in this Cell Data Bank. And we are looking at whether we can predict a risk of hospitalization for people who have been formally diagnosed with dementia in 6 months' time. So there's condition on they are positive diagnosis already. It's not just any patient. That's important because that class imbalancing problem will be very different. And we are using the model very simplistic. We are only asking feed forward a neural network to perform the prediction. We make some incremental changes to the neural network in that we wish the network tell us a bit more about the features that have been used for prediction. So very minor changes to that. So we used a penalty function trying to force the first layer of edges, the weights we have computed, to be two extremes, to binarize them, to have as sparse as possible, based on assumption that 0, of course, is going to be irrelevant, but anything close to 0 in terms of magnitude, we consider as less important compared to larger weights. Now, that's debatable. I understand this is not necessarily true, but to some degree, that can be assumed. So just based on that minor modifications, and we can see reasonable prediction later on. And that derives 3 different categories of features, and all of those are, in conversation with clinicians, they seem to be all relevant. And useful thing about this is, in fact, now we can quantify them, given a very large chunk of feature factors that we started with. So I'm going to quickly go through the slides. Apologies, they do seem very different, and so I'm being a bit lazy here, just using my students' slides for the ease of that. Well, these statistics we see all the time in terms of dementia research, for example. We know this is a huge problem, and it is a multifaceted problem. And what we're presenting here only touches the surface of the small issue that's related to the dementia. And fundamentally, we're interested in how we predict risk of institutionalization, because the care for the patients who can independently manage themselves compared to somebody who need institutional care, the cost and the resources are very different. So needs are very different. That implication on the cost are also very different. So if any forward-looking prediction would be useful in terms of patient management and resource allocation as well. So what we concern is finding those high-risk individuals based on possibly risk factors, which is already being indicated in the literature, but we want to find out a bit more whether they are reliable and to what degree they're reliable, and also explore whether there are additional risk factors that's not being looked at that can be useful in predictive models. And if you do look at the literature, what sort of things being done, there are various different scale studies, and many of them, I would say majority of them are quite small. So if we count these ones, you can look at hundreds or at best thousands of different patients data sets. So it's quite small scale of data. And part of the challenge is really connect as exhaustive as possible in order to build the predictive models. There are some much larger studies like these two, comes to tens of thousands of data samples. And they do interestingly point out some of the factors that are to do with medications and to do with how their prior mobility issues they have linked to the dementia, which is well understood. But there's quite a bit of space that further research is needed, really. As I said, the method that we're looking at is very basic. So we only look at feed-forward neural network. We're looking at how we can train them more efficiently. So we're using a snapshot ensemble, a method which is being reported in the literature, and with minor modifications in terms of how to make the first layer weights to be sparse as possible. So then we can actually derive features in that way. Then I have to say there are many different ways you can derive the features. It doesn't have to be this way. This is one of the early attempts we have, and I think it'll give some indication what sort of feature we can derive just by using a very basic model. So we understand neural network very well, and I said already that the ability of transforming linear inputs to non-linearly, then followed by linear combination, and do that sequentially, give you the ability to predict something rather complex. The problem inherently is how do we entangle all that and link back to the individual features that you put into the system and start with. So what we're saying is, let's take a look at the first layer inputs. Can we make these ones sparse as possible? So in this attempt, it's really basic. We're saying that let's put a regulation term on the first layer of the weights, and we want the distribution to be as extreme as possible. So there's no guarantee, of course, you have binary. In reality, of course, you never have binary, but it does push lots of the weights close to 0, that indicating that we can reduce them to a certain extent. There's notable difference to PCA, for example, linear analysis. We know that because it doesn't have the nonlinearity and doesn't based on the variance and so on. So it is different to unsupervised way to reduce the feature sectors and so on. Right, so I didn't mention snapshot ensemble purely because that's the computationally list gives a very efficient way to calculate the weights and so on. But you can use different ones to do that. The basic idea is very simple. You train that once and you perturb along the way given certain parameters, and you record where you have the local minimums, and you've taken those ones as local models and then ensemble them together to produce a final model. So what are the things we can do with the weights once they converged? Well, we could look at basic statistics of these feature weights in the first layer. I said, based on the assumption that if the weights are zero, simply mean that connections haven't been used at all. And if the weights are the magnitude close to zero, mean that that feature perhaps, most probably, that is not that important. So then we can rank the feature that way. And that's the basic assumption we use. And let's see how that lead to us. So we're using just over 100 years of data records we have in the system. We end up just under 60,000 patients, and that gives us a total over 52 million individual events. So it's rather big data sets that we're dealing with here. An event code, so original feature space is 55, more or less 55,000 events. So it's quite a large system. And you can imagine the sparsity in all that. And also individual patients have very different sequence to each other. So you could have patients have very dense record of those electronic record of the events happening in that long period, and you could have individuals have very few. The part of, in our case, we said we're predicting 6 months in time where the hospitalization happened. So that considered part of negative ones. We have to match those ones. So we find those ones which have similar demographic informations, that have similar records matched in other areas, to consider as negative, so it makes them comparable. So we end up, these are statistics, the table here showing what sort of population we're looking at. Okay, relatively the gender balanced in terms of for this particular dementia data set. The coding we use is ICD-10, is international coding standards. I think there may be a new one now, ICD-11, I'm not sure. ICD-10 is still used in NHS, certainly. So I said that's a condition for positive and negative, so make them relatively comparable between the two sets. And then encoding of that is straightforward. You're just taking that as 1D vector, and then you have those events encoding the space, and you can classify and such. Very much standard classification, validation, and testing procedure. Nothing really new here. We are comparing against standard, very classical approach like random forest. One of the things that you can look at is evaluation very based on feature evaluation very clearly, and other things which are used in health informatics domain very often mainstream as well. So what sort of thing we get? Well, as I said, we have these one highlighted here, which shows a much higher importance in terms of prediction. And all these are highly relevant. So we don't have any big surprises here. It does show, for example, nightmares, the frequency of nightmares, essential hypertension. These are strong indications of severity of the dementia, and they are pop out in these highly ranked features. And certain medications also is a clear indication of the severity of the dementia. And then you have other procedural events. Interestingly, social group economic background plays a role in the predictive model to predict whether it's severe dementia or not. And these just simple visualization to show what sort of sparsity we have in the feature side. So that's pretty basic in terms of methodology. And if we do compare to some of the very classical and perhaps basic techniques, we see some improvements from this new network model. So there's certainly scope to work on. We do have far more sophisticated methods applied to this, but the improvements so far we see are marginal. It is better than the proposed new network we have here, but I thought it doesn't worth to mention those, given that the complexity that's in other models we have used and developed compared to this very simple model that we're showing here, that's showing the difference between the two. Right, I think that's the first case study on the dementia side I have here. I don't know if any comments, before I jump back to the second one.
Sure, can I ask a question?
Yeah, sure, of course.
We also have done something like that similarly in our team as well. We try to use patient records to predict schizophrenia type of mental diseases. So so you have these importancy values, right? They basically make your network a bit more explainable and interpretable. So I'm just wondering if you have observed any bias in the algorithms or in the data that this kind of importance kind of show. Say, for example, in our system, we found that the gender has a large importance, but gender has no medical values to the schizophrenia condition that we are observing, right? It's just a statistical bias. So did you observe something similar?
Yeah, certainly. I mean, that lots of these examples we can find. So in this case, not in particular this case, gender is an issue because data itself is rather gender balanced. But as you know, there are for many, if not majority of medical conditions, age is a predominant factor, risk factor. The older you are, the more likely you're going to be dementic. And the older you are, you're more likely to suffer from cardiac conditions and so on and so forth. So that's intrinsically, that's a dominant factor. And we also see race, for example, we see in the second example we show you that it does show that as an example of possibly that triggers in those the prediction in the model and overemphasize on those ones. So what we did is we analyze them first to see what come up, and often we delete those ones in the model and see how model converges and see the difference between the two. I'll be interested to know what other ways you can tackle those obvious bias in the system, apart from preconditional data and explicitly, essentially, a Bayesian study we're looking at removing those ones and see the stability of the model, the performance, the predictability, and repeatability. So then understand those bias, the role of those bias in the predictive model. But any other techniques I'll be interested to learn. But that's the sort of thing we do when we encounter those. But certainly that's an issue. Yeah,
it's a very challenging problem because we also try to filter away some of the obvious bias, but there might be some kind of intrinsic bias that we are not noticed. So it's still an ongoing thing, I think.
Yeah, certainly. I mean, some of that is better understand in terms of how what role they play. You can clinically explain them, like for example, said age. If you see that and it's a good indicator of the outcome, and then we know the reason behind it, and some of them less obvious. So yeah, I think ablation study may be a good way to understand those as well. But yeah, it is a challenging one to deal with, the intrinsic bias in the data set. Yeah.
All right.
Thanks. Okay, great. Okay. Right, I'm going to move on to the next one. So this is the case study looking at sepsis prediction. Slight difference to what we had before. The previous one, relatively balanced in terms of in positive and negative, because of conditional on those had dementia already. The lot of follow-on work is ongoing at the moment. So for example, we're only predicting whether you're being hospitalized or not in 6 months' time. But we're not telling the details of that because there's a huge difference between the trip to A&E and compared to have a long stay as an inpatient. And in reality, that's dementia patients you're looking at any time of the day in the UK, about a third of the hospital beds are occupied by patients with dementia. So not necessarily because dementia in the hospital, but the patients does have dementia as one of the condition. So that has huge implication on that. So we are working on different ways to more granular approach to this. Then this one here, septic, is also very relevant, but it's different in the sense that the it's hugely imbalanced data sets we're dealing with. We're dealing with a large chunk of negative with small positive data sets. And this creates a huge problem for us in terms of class imbalance. Another thing is similar to the dementia data set we have, but this one is more so, is that the sparsity of the data. Because of previously conditioned on positive cases, so you can expect a certain homogeneity among the patients. They will have memory tests done, each one of them. For example, they will most likely go through certain clinical events, apart from their different comorbidity issues. But here, the patients are very different to one to another. And we're looking at negative case where people don't have sepsis at all, even though they are ICU patients. So it's much broader cohort of patients. And we are using LSTM model as baseline. So I think all of us are familiar with those, so we're not going to go through that. We are using some of the principles that we developed for the previous model and applying here, but devised slightly differently. For example, we are dealing with high imbalance problems. So what we can do, well, we can perform cascading, for example, to eliminate stage by stage, what are the negatives. And also we're looking at changing the cost function slightly to impose different level constraints in order to derive those eliminate different level of negatives. We're also looking at other things. For example, once we're predicting as a positive case, because it's a time sequential case, you don't want to reverse the prediction in the future to say, oh, the septic can now become non-septic. It doesn't make any sense, clinical sense. So we do a few modifications of those. I'm not going to, in the presentation, I'm not going to go details LSTM and cascading, for example. And that's simply, I assume everybody very familiar with anyway. So that derive certain results, which I can explain a little bit more later on. That seems to be working well, but also there's tons of work we can do as well. So I'm just going to quickly go through the slides here on the septic. So before we go into detail, this is useful to know that these three different categories are very indicative in terms of diagnosing sepsis. Blood circulation, that's very important, the cardiac functions, and then respiratory circulation, so how well the breathe and oxygen delivered to blood circulation system, that's really important. And then you also have the third one, essentially how was the mental ability to respond to the environment, so the level of consciousness. These are the key things that clinicians use to diagnose whether the patient going through septic shock or not. Now the difficulty, however, there's no single biomarkers that tells you that. There's a combination of factors that are taking place, and then also you consider patients' other comorbidity issues. So you have patients could have respiratory system problem already. Now how do you differentiate that? It's because of the septic shock, or is actually because the person has a comorbidity in that area. So that's a huge challenge on those. There's a different statistic to highlight the importance of work in that area and lack of timely diagnosis of sepsis is what leads to. But one statistics I just want to point you to, not many people have this, is that it's depends on which hospital or health boards you're looking at. And then statistically, you look at the UK, you're looking at between 4 to 8% increase of mortality every hour of delayed treatment for sepsis. So if you can't diagnose them, there's no way you can treat them. So give them broad spectrum antibiotics to treat the septic shock. So here you're looking at, take an average, let's say 5%. If you delay them for 24 hours, then the chance of survival is very, very slim. Right, so we are using two data sets to evaluate the methods we are looking at. First one is a PhysioNet, which is public, both are publicly available. It's a challenger data set, and if anybody interested in can have a look. This is smaller between the two we use. They are more sort of cleaner set up. The data has been carefully created. There are large variations between the training set and the testing set, however, particularly the set they used for ranking different methods, but that's perhaps a separate issue. So we're looking at 40,000 or so patients, and genders are relatively balanced. Average age just above 60, the positive case rate is about 7.3. So it is quite unbalanced data set to deal with here. The other one is also very popular as well, but contains a lot more patient data, but also less organized as well. So you're looking at MIMIC-3, you have 53,000 patients in the data set. You're looking at 12,000 or more than 12,000 medical events based on previous ICD-9. So this is a lot higher feature space than the PhysioNet one. Interestingly, the male and the female divide is the same. The 55 versus 44 is the same as the previous ones by coincidence. The positive rate is 4.2, which is slightly lower. So the imbalance issue is even more severe in this case. Apart from the usual way we evaluate this system, there's also the so-called utility score, which measures how useful that particular model performs. And in this case, we said optimal prediction is 6 hours in advance. So you have, let's say, this is optimal time, then at that point, you have the highest reward for positive prediction. Anytime earlier, you have lower rewards. Anytime later, you have lower as well. And when you reach the stage where passing the prediction time, for example, 3 hours afterwards, you don't have any reward whatsoever. Same thing happens for the negative. You can't see, then it's not exactly zero. So you have slight cost involved in rewarding in terms of positive prediction, but any negative prediction, false negative, you will have large costs if you put in the positive. So this is entirely handcrafted. There are some issue with the negative predictions. So that's explains some of the data we see later on, the discrepancy between utility score and normal metrics we use. So I'm not going to details of the cascading LSTM and how we deal with the margin hinge loss to impose them, and also the idea of switching back and forth in the prediction. So negative reversal penalty function, and these are again very small changes to what a standard LSTM would do, and none of those are dramatic changes to the literature. How do ensemble classify together to make stronger predictions? So really, we're dealing with the class imbalancing problem and the sparsity in the data side. One thing I don't have time to talk about, which is something that we are working on at the moment, but everybody in this space been struggle with, is imputation problem. So what do you do with data which doesn't have record? And this is a huge issue in terms of sparsity. There are ways perhaps you can use generative models to synthesize and so on and so forth. But what we're doing here is just simply using demographic data and then impute them using the average observations we have. So what's the results we have achieved here? We're looking at reasonable in terms of informatics side. If you are working computer vision image processing, you will see these numbers are rather low. You're looking at 50% also true positive rate on just about 50%. Set B is the training, set A is the testing we have here. So you have just about 50% for the training set, and just below that for the testing one. True negative rate is higher, but remember the negative samples rate numbers are far greater than the positive ones. So you end up F1 score is 0.4142. These are the numbers we're looking at. So not particularly exciting in terms of absolute values. How that compares to other sort of methods in the challenger data sets, we are doing reasonably well. One thing I pointed out earlier is that the discrepancy between utility score and some of the other things that we commonly use. For example, Oracle, we look at the area under the ROC, you see that the second method with a high utility score and scored a very, very low, negligible amounts in the area, which is a very strange way. So that indicating we need to look at the utility score function again, how to evaluate the system. Similar things observed in mixed data sets. We do slightly better overall with many other methods as well. So there are different configuration LSTM we're looking at. So ours close to 70%, 0.69, and F1 score is higher, marginally higher some of the other configurations. But still, I said that there are areas we need to look into. Interestingly, perhaps look at random forest again. It's a very basic technique and it performs rather well in comparison. So it does tell us that, yes, we can look at complex models, but how much gain we have and what we do in terms of interpretability, because random forest can give you feature ranking very easily. So that's something that we are looking at the moment. Well, speaking which, that's one way we could do to improve how to interpret the model is by, let's say, have some linear layers in the system and then looking at how well we can unlock those features. Well, in the first example I give you, we said that let's force the distribution of the weights in the first layer to extreme as possible. And we can apply the same principle we here. So we said we have LSTM, cascade together, build a model, and then doing the prediction. And we have large feature vectors to feed into the system to do that. Well, what we could do is borrowing the same idea we had before, is that let's have one by one convolution filters. And each of those is nothing more than just examine the linear combination in the feature dimension, right? So each one there's an entire linear combination. So we could do a few of those, look at a few different combinations and see how well those would do feed into the LSTM. And it turns out they can perform very well in comparison. But the benefit of that is that we now have a matrix of, if I use 5 of them and input the features 12,000, I have matrix 12,000 by 5, and that allows me to actually analyze the feature set based on the weights, and that gives me indication about what features are useful and important in predictive models. We can do a little bit more, say, let's force certain distribution of those, again, impose sparsity. So before we have the entropy minimization to impose the sparsity of the weights, we can use this parametric function here to impose the sparsity of this coefficient weights. We can do different ways, but that's one way we tried. And that actually turns out very interesting. So if you look at the visualization of this coefficient matrix, white means close to 0. And the histogram just shows the majority of them are really not useful in predicting whatever the outcome would be. And only a few of them, in comparison, the number we have 15,000 also actually useful in predictions. So then we can look at those ones based on those statistics, and then you can derive those features, and also you can look at the average and standard deviation indication about how useful and robust they in the predictive models. If we keep 850 or so features from 15,000, you still have very comparable performance, but then you can interrogate them. Now, some of them very much relevant, heart rhythm, cardiac murmur, they're indicating what's going on in the cardiovascular system. But some of the more useful ones somehow embedded later. And another observation we see that the tails down very quickly. So the bottom end of those 800 or so features, they are really, really close to 0. So that is indication that why not do another round? So that's exactly what we did. So let's take another round, look at those ones to reduce a bit further. We sacrifice 3% also in terms area on the curve, but that gives you more interesting results. And now more relevant features are here. For example, this one's really interesting. You have verbal response. Remember we had 3 categories: cardiovascular, respiratory system, and then the response to the clinicians. So the verbal response are one of the top ones now, and we still have the cardiovascular and also respiratory system indicators captured as well. So that's interesting. So you do some very basic linear layer, insert linear layers into slightly more complex model as we had before, and you can actually derive features which are indicating what are important, what's less so. Right. Before I jump back to the main presentation, any questions for this part or any comments?
Hi. Can I ask a question?
Sure, of course, yeah. Sure.
Yeah, I'm also dealing with the highly imbalanced long-term time series data. So I think maybe I can borrow some your design from your training. So but I'm actually curious about what is the actual length of your data? You mentioned you use 6 hours data, but it's contains every second data or every minute data or something.
So data is collected on hourly basis, so every hour.
Oh,
So they are laboratory testing data, and then they are in the ICU environment, they're collecting those data as well. So that's what in the challenge data sets and also in the WMIX data set, I think, on hourly basis. Yeah.
thank you.
So in terms of the imbalance issue, yes, I think there are. I mean, we are using weights, for example. We tried different ways. Which, you can increase the weight for positive samples, but that's same as duplicated sample. There's nothing different between that, and you will increase the bias towards the sample as well. We try the perturbation of those data sets. So you have positive samples, you can perturb them, and cascaded ones are actually quite useful. You can partition the space of the negative ones. You partition them quickly, and then you can derive a more meaningful partition between positive and negative. So clustering the negative space, we find that's useful as well. But in biology, it's a difficult issue to deal with for the imbalancing issue. We have to make an assumption that the positive samples are indeed representative. If they are, to the extent that the positive samples are not sufficient in representation of distribution, then very little we can do. So all the things I mentioned, whether you analyze in the negative sample space first, and then you're starting to perturb them, generate more data samples, adding more weights to them, it's all based on assumption that the positive samples are covering enough of those. But yeah, I think reinforcement learning is another thing we tried. How to effectively carve out a space. That also is interesting in terms of work out the imbalance issue as well.
Very helpful. Many thanks.
Thank you. Yeah. Okay. Right. I know I'm run out of time. I could stop here. I lost control. Yeah. Okay. I know in the abstracts I cover graph deep learning as well. I could stop here. I could take 2 minutes, just quickly go through them, and then we leave a little bit of time for questions. Is that okay? Yeah. Okay. Great. Okay. So the apologies for if you are interested in deep models. You don't see much deep models per se, but hopefully give you some indication what you can do on the health data side. We do perhaps a bit more serious deep learning as well. I would love to tell you some of the very interesting discoveries we made in terms of preserving side, specifically federated learning in deep learning. It's well documented recently. We had a paper on that as well to show that federated learning can actually have a serious gradient leakage problem, and we derive a deep model can recover much better than existing literature reported there. But more interesting, perhaps, if you find a way actually to prevent that leakage happens, at least to minimize the risk. Maybe that's a different occasion. But we do deep learning on the theoretical side, also application side. I'm going to mention just very briefly that's some of the early attempts we did, because we're talking medical application. Also, this will be interesting just to mention that using graph learning applied to medical images. So I mentioned that we're dealing with large, dense voxel grid. Is it really necessary computation taking place in a voxel grid? What happens if I reconstruct the data that in a lot sparse grid, and you can represent in a graph, for example, and then we can use graph deep learning to do that segmentation if that's of interest. And if anybody interested in the graph deep learning, you can perhaps check out the survey paper written by our students lately to see what sort of different techniques and approach already and what are the challenges. What two things we're really looking at in the graph deep learning is that how do we translate what's something taken for granted in the spatial domain in the standard setting? So we do convolution straightforward. You have the Cartesian grid, that's just linear computation. Pooling straightforward, you downsampling, that's what you need to do. But what do you do that's in the graph in the unstructured environment? So what is neighbors to start with? How do you define convolution? How do you reduce the graph representation? So these are more challenging in that domain. There are a few different ways to do that. This is one of the methods my PhD student proposed in terms to transform the graph representation in, apply Fourier transform, and then you define this in the spectral domain, define the convolution and multiplication of that, and then you have control points to manipulate that distribution. And then you do an inverse transform to the original graph space, then you give the convolution. Now this is reasonably well, and then it's more problematic in a sense is the pooling side. So you have a structure, for example, very dense with grids, and then you want to sparse them over time to the pooling. Now here is more problematic. We use, for example, geometrical methods, just looking at purely on the grid side, discarding data and to downsample them. But there's issue of connectivity, how to handle topological changes as well is very difficult. But even you make some compromises, you make some assumptions and make some compromises, it does show that you can transform some of the segmentation problem, which may be very computationally expensive, and make that much more efficient. So here's a very simple pipeline. They're saying that we take in medical data into the graph, transform it into graph learning problem, and then we perform convolutions and pooling in the way I just briefly described, and then you do usual way of the CM. And what it gives you, so in the medical example I said earlier, is that give you a reasonably efficient classification segmentation of the geometry. So this is an example of full 3D torso of the person, and that number of candidates you have to evaluate is much bigger. So if I have 512 by 512, I said earlier on, is you're looking at 6 to the power of 15. This is enormous space you want to evaluate. Of course, you can do approximation, for example. Instead of fully 3D evaluation, you can marginalize the distribution, and then you can do sequential steps instead of evaluating in one go into position, orientation, and scale, and so on. You can sequentially doing those to reduce a bit of those computation. Right, this bit is less interesting. Once we've solved the mathematical elements of how we actually perform pooling and convolution, and then becomes more a software engineering challenge, how to assemble it together. And unfortunately, I don't have anything more exciting to tell you apart from large elements of trial and error, with students polling through for a long time on the GPUs, trying to work out what's best architecture. I think this is one area we all have to look at, how to optimize that process. So just quickly show you some of the results. I know we are over time. We do reasonably well. So we have good sort of mesh accuracy and less have outliers into deviation to the ground truths we have in terms for segmentation. But that's one area we are working on in terms of segmentation and combining with graph deep learning. So I think I should stop here. I used the full hour already. I know everybody's very busy, so I should pause here. If any questions, I'm happy to take them.
All right, thank you very much. Any questions?
Sorry, can I ask more question about the segmentation?
Yeah, sure.
Yeah, so I'm not familiar with the engineer force. So can I understand it's like a snake algorithm, and you need to update your parameters in each iteration when you influence. Need you? Oh.
Yes, you do need to update the parameters for the representation, yes. For the semi-implicit one I showed.
So how is your actual inference time? So can it really accomplish real-time inference as you mentioned?
Yeah, so the method I showed you, and in one example there, you can converge that very quickly in just very few iterations. That depends on a few things, of course. It depends on how robust the representation is, whether you can handle large deformations, and also, of course, depends on how powerful your classifier is. But with the method I'll show you, they can converge it very quickly as well. Yeah, there's a code available online. If you're interested in, you can just go and download and give it a try.
Yeah.
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 Any more questions? Right. So shall we thank Professor Xie once more, and thanks a lot for the interesting talk. Actually, there are quite a lot of people here working in the healthcare related or biomedical engineering related research using machine learning and computer vision algorithms. So I'm sure your talk will be interesting to them. So if they have any further question, I'll just forward their request to you by email. Yeah, it's a bit unfortunate that we have another departmental meeting just following this one. So some of them need to leave earlier. Yeah.
No, I totally understand. Yes, great. Okay.
Thank you very much for your talk, and let's keep in touch and see what we can explore doing together in the future.
Yeah, thank you. Thank you, everyone. Bye-bye.
Thank you. Bye.